name is Jiang. I'm at Georgia State University in the Department of Mathematics and Statistics. But all my degrees are from physics. And sometimes introduce myself as a physicist sitting in a math department, doing biology, using computer science tools. So anyways, here I am. Thank you for arranging the talk. And learning from Alexander here that because I'm going to be here till December, I'm trying to go shallow and broad than on specific subjects such that we can have more time to show you what I have, what I'm interested in, and then we can have more opportunities to talk and go deeper. So the main story today is on collective cancer elimination, and we all know that cancer tumors don't kill, it's the metastasis that kills. And in order to kill, to establish secondary growth, the tumor cells will have to break away from the primary site and then travel to distal part of the body, the brain, the bone, the lung, the liver, and kill the vital organ, essentially, by killing the patient, by killing the vital organ. We all heard of someone, know someone who's got cancer, and it seems like the killing rate of cancer is higher and higher as the time goes. And we tend to think of cancer cells as this very invincible entity, right? They break away, they establish second growth, they kill the patient. But the fact is, the cells breaking away from the primary site, 99.99% of them will die because of the host defense system. They really have to run the gauntlet in order to be able to establish the secondary growth. And in that context, it's easier to understand, or it's easier to conceive the concept that if you are the loner trying to invade a homeostatic site in order to establish secondary growth, you're an invasive species, and a loner is much harder than if you bring a small team with you, right? And especially if that team, some are good at growing, some are good at migrating, some are probably good at scaling the wall, and some are good at killing themselves, right? If you bring a small team with you, your chance of success will be much higher. So here's a spheroid from breast cancer from Friedl's work years ago, that it shows the collective strength, invasion strength breaking off from the big spheroid tumor. And that looks like there's the tip and the follower, right? Some kind of differences, heterogeneity between the different cells coming out. And you can see the tips not in this picture, but the tip cells are good at degrading collagen. They are very good at migrating. The following cells are actually good at proliferating. Not good at migrating. And in this kind of a heterogeneous mixture, you can start to think about social interactions, division of labor, symbiosis, collaboration, collective decision making. All those terms that are good for collective entities, heterogeneous entities, or ecosystems may become relevant. Or the theoretical models or the tools that we use to study those systems may be borrowed in this context. Back in the 1971, Nixon declared war on cancer. Half a century later, we're still looking for the magic bullet that tries to find and kill the driving cells in tumor and not harming the host too much, right? So that efforts largely failed so far. Gatenby proposed this idea of managing cancer as a chronic condition. And that is actually borrowed from the ecosystem again, that history is rich in examples of managing the invasive species while keeping the whole metastable. Managing the invasive species to a small portion, its influence of the whole ecosystem is much less disruptive than trying to find and remove all the invasive species. That's very difficult to do.
So in order to put such a strategy to work, we really need to understand how the cancers invade of the egg surrounding tissue and be able to maybe trim off this invasion if possible, right? So here's the my collaborator Adam Marcus at Emory's work. They put non-small cell lung cancer cells, spheroids, non-small cell lung cancer cell spheroids. These are primary tumor from patients, so it's heterogeneous intrinsically. And some cells come out in time, and it's embedded in collagen. So some cells come out as individual cells, some come out as a collective strain. And this collective strain is led by a leader and followed by the followers. And you can see in this example that the leaders falls off, it walks so fast, and the rest just won't go. So this separation of labor here.
This is in vitro, right?
I'm sorry.
In vitro.
In vitro, yes, it's embedded in collagen.
Okay.
And I'll have a color code here. All the black references are not my work. Only the red ones are going to be work from our lab. So in this work, oh, I just told you, the leaders break off and the followers just don't go, right? So that's a clear leader and follower. And in here, there's actually a rare altruistic leader that it breaks off and then comes back and then grab the rest and come out. We don't know how, we don't know why. And they designed this new technology. They transfected all the cells. Well, they make all the cells green, and then they transfected it with Dendra, which is a new class of photosensitive molecule that when you shine that laser on it, you can turn it to red. So now when it comes out, you shine a laser on the tip of all those protruding fingers, and then you can sort them out. Right now you have all the leaders, and then you can collect all the followers. So now you can grow leaders and followers separately and analyze them separately or mix them if you want to. And that they show that in that work, they show the leaders and followers are very different genetically. The leader is a high VEGF signaling, low in photoadhesion, low in proliferation. And the leader is also high in this JAK1. JAK1 is delta, similar to delta in this not delta interaction. Okay, I'll come back to that later. And also the leaders have a lot of dimension, which means they might be more like mesenchymal cells than epithelial cells. The followers are more epithelial-like. The follow leaders also high in VE-cadherin and VEGF-R, VEGF receptor with VEGF signaling. And I don't need to tell you which one is all leaders and which one is all followers. The leaders just migrate, they don't proliferate very much, and the followers just grow, they don't migrate. So no collective invasion in pure culture. And if you add some followers to leaders, it actually helps the leader proliferate. Or you can add some of the medium that followers been growing and to the the leader grows back. So followers secreting something that helps the leader grow. And the leader, if you add leaders to the follower population, they start to collectively migrate. If you add medium from leader to follower, it inhibits the growth. Right, so complicated story.
Yes. Can you just repeat? So the followers secrete something,
helps the leader grow. The leader secretes something, inhibits the follower growth. But the leader also helps the follower go out, such that they have more access to nutrients, etc. Right? So all this is summarized in this paper. Oops. In this paper, in such a interaction network, the leaders secrete fibronectin such that they can migrate. The leaders are high in VEGF-R, and they have this VEGF-based signaling. In fact, that signaling is not delta as well. I'll come back to that. And then the leaders inhibit the follower growth, the follower promotes leader growth. All those things add together. And Seth Haney from Alex lab at UCSD built this model leader, follower population, leader inhibits follower, they have growth competition, and then follower promotes the leader growth. Leader extrovert mediated signaling help the leader moving out. The leader also secretes fibronectin, such it helps the cell moving outward. And all this set into a set of ODEs, and the interesting parameters are those feedbacks. That's feedback. And like all the mathematicians or physicists, you can do dynamic system analysis for different domains, parameter regions, such different dynamics exist. They can either can extinguish, they can coexist, they can have explosive tumor growth. They can also have treatments added. Chemotherapy alone kills some of the tumors, but if you also, instead of trying to kill the tumors, you target those feedback, mutualistic feedbacks, you can have the tumor really removed much better. So all this network.
So can you?
Yeah.
You might go back. So you have put parasitic mutually. So what is the definition of you parasitic?
Parasitic. One relies completely on the other for growth. And leader parasitic, that's the follower parasitic. So it relies on the feedback growth.
In your pair equations, right?
Oh, that's not my equations. Set.
So how accurately can these parameters be measured? They cannot, right?
No, yeah, there's no, we don't know what factors are involved, we don't know, so this is a purely theoretical exercise. So this is another purely experimental exercise, is we treat leader and follower as these games, and leader benefits follower, follower inhibits, no, follower benefits leader, leader inhibits the follower, leader grow lower than follower, but there's also a possibility of follower could turn into a leader, and leader migration promotes the follower. So in this, we can write out the replicator equation and do other things too. People here all heard of several hospitals. Okay, quickly, we divide the domain, divide space into lattice, three-dimensional lattice, and regions of domains called a cell, maps to a cell. And the numbers on this domains is called the cell ID. And the cells can interact through a surface interaction, type dependent surface interaction. So the cell number 3 can be an endothelial cell, and the cell number 2 can be a leader cell. Okay, cell number 3 and number 4 can be a follower cell. And we assign them different surface properties such that this time-dependent interface energy describes its differential emission at the cell boundary. And the cells also is constrained to a target volume elastically. And this target volume itself can grow and shrink for cell growth and death. And also the cells can respond to an external signal. It's a way to bias the boundary movement such that it can describe chemotaxis and can describe urotaxis, any other external field described by this C. And the whole pattern update is Metropolis algorithm. And in addition to all this, you can have cells carry receptors or intracellular genes or signaling network inside, and they do calculations and to decide what they want, how they should behave and what they want to become. And then outside, you can have this PD of oxygen, nutrient being diffusing and being consumed and secreted, or also possibly interact. So this is the multi-scale cellular pulse model, we've used it to describe this solid tumor growth in different environment. We used it to describe angiogenesis, tumor-induced angiogenesis. So the VEGF sets up a gradient. The tumor in here, hypoxic tumor sets up a VEGF gradient such that the endothelial cell will grow toward the higher concentration of VEGF, and we have the blood vessel grow into the tumor in the ovarian cancer relapse growth, and then this kind of blood vessel, leaky blood vessel, can also be used in describing drug delivery, whether it's intravenous, the systemic injection or intraperitoneal injection. I want to draw to your attention that the similarities, this kind of a protruding finger led by a tip cell followed by the follower cells or strength cells that look angiogenesis that look very much like the collective concatenation. In fact, it's not just the shape that looks like it. In angiogenesis, it's VEGF mediated Notch delta and metal inhibition that forms this tip stalk phenotype differences. And so it's not just the morphology, dynamics, and also the signaling. There's a lot of similarity between this collective invasion and sprouting angiogenesis. So back to the pulse model. Here's a very minimal model. Leader green, follower blue, and all I need is that leaders migrate. I set up a gradient such that the leader would want to go out, and the followers want to stick to the leader. That's all. And we'll have collective invasion. And I can have the leader in some simulations, leader walk a little too fast, and the followers won't be able to extend. And in some cases, the two leaders just run into each other, but because leaders don't adhere to each other very strongly, you end up with only one leader in each stand. And here's only leaders versus only followers, right? So I can have a very minimal condition for collective invasion. Now, what about all the other interactions? How do they do? What's the purpose of the growth inhibition, growth promotion? So my student is working this summer to explore all those parameters.
Sorry, I was just wondering, the previous simulation where you had all those case scenarios, were they always the same set of parameters? But
Yeah.
different initial
Yeah.
conditions?
Yes, yeah. So I can just cut open the spheroid now. It's looking at the spheroid front, right? It's very tempting to think of it as a fluid flow problem. Does look like it. Well, the first impression was it looks like almost like the front stability of PDC equation and whatnot. So we tried to learn the governing equation just from, because I can do lots and lots of simulations. I can do machine learning. I can do neural networks. The sparse regression simply package didn't work because they are good at ODEs, right? They're not very good at PD. We have a collaboration at Georgia Tech for this ident or weak ident that effectively give me 6th order polynomial fit for the front, which is not good. So we're now looking on physics informed neural networks with the terms like PPC, with terms from Navier-Stokes, and we're trying to learn those coefficients. It looks promising. So maybe in a couple of months.
The 6th order polynomial you dislike because
there's no physics in 6th order polynomial.
okay. But those machine learning techniques you get out.
Right. But that's because all the gradient terms were not included. Well, we included limited gradient terms such that those terms come up to be small. The coefficients of now we have a much enriched library of more physically relevant terms. And we're hoping that could be the case.
What are you using as a summary statistics?
Comparison of the shapes. Yeah. The interface is right now we are just putting on top of each other.
And then
but-
average all the interfaces.
Right. And I can show you different simulations. This is with fewer leader cells. This is with more leader cells. And just by eyeballing it, they seem to have some kind of a life scale selection. And that indeed turned out to be true. We can measure the invasive area being the inscribed area and the outer area. This in the middle is the invasive area as a function of the leaders and the function of time and the number of stalks converge. And that if we have a lot of these kind of leaders break off or little defectors break off, they interrupt the collective invasion, such that you actually end up with lower invasive area over time. So we're still working on it as of this summer. And
Just
Yeah.
a question. So I would imagine that if the leader goes too fast,
they break off.
then it would be convenient for the leader to just wait for the follower to remain close, no? So I wonder, maybe, I mean, why don't this, maybe the cells do that? I mean, so that's.
The altruistic ones that did it, right? We didn't know why. We don't know how often they are. That's very rare. In our simulations, because we want the leaders to lead the migration, so they always have the driving force of moving away.
No, yeah, but I'm just saying, why not thinking that, like some of the ants move in tandem, so there is the leader and the learner, but then they wait sometimes for the learner to go to show the new nest. So maybe they should do the same, some way of slowing down instead of just going, oh, I'm going.
Possibly. We can always change the rules, but experimentally, you have the cells. Okay, so this was trying to simulate the lung cancer spheroids. And then they also did the breast cancer spheroids. And they tracked the individual ones versus the leader versus the follower. In our simulation, the individuals are leaders breaking off. But in further analysis, that the individuals are genetically even further away from the leaders and followers than between followers and leaders. So it seemed like there's a possibility that individuals are really the truly selfish ones.
Yeah, so in experiment, do they see lots of breaking off of leaders?
Some,
So what's the difference? Do they have a number? I mean,
depending on the different cells we use.
pick a cell.
Pick a cell, right?
Yeah.
41. There's a lot more than lung cells.
And when you say a lot more, so it's majority, really large majority.
I don't know the numbers.
Okay, so maybe it's a real,
Yes.
there is a real mechanism that they just wait for the follower,
Okay.
if it's a number exact.
Okay, we'll talk about that. There's a number game there too. So this is okay. I need to hurry up a little. So we have a minimum sufficient condition for collective emission, and we're trying to answer. What about the others? Are they trying to make it more sensitive, make it more robust, make it all the other interactions? Are they trying to make it more stable, the collective emission? And it's possible to manage collective emission by interrupting the social interaction instead of trying to kill the tumor. And it's possible, we don't know yet, to learn the governing equation such that we can be truly predictive of the collective cancer invasion. So the second piece of the story is looking outside the tumor, outside the cells. It's the extracellular matrix around the tumor spheroid. So the tumor spheroid here now is in the dark region. At 0th hour and at 24 hours later, you see because of the cell migration, the collagen now outside become more aligned. This is second harmonic imaging of collagen outside. And for 1, 2, 3, 4, this is this. I don't know. So maybe 5 different cells after time. You can see
Sorry, can you just
different
clarify?
degrees.
So you
Yeah.
say aligned to what? You mentioned.
When they are aligned, they are brighter. The collagen extracellular matrix network
Yeah.
outside the cell,
Yeah.
as the cells try to move outwards, they remodel the matrix such that the structure is different. We can see the difference between this one. is the top is at the 0th hour and the bottom at 24th hour.
And you can say align because they sort of seem straight.
They sort of think straight, and you can also segment it, the fibers, and then just measure their alignment. And then you can make a alignment field, alignment map. And you can then compare this alignment field between different experiments, different strains, different mutations. And that can be used as an independent marker for quantifying invasiveness. So that's sort of a visual of how invasive these cells have been, because the cells migrate by tugging on the fibers, right? They have to really walk on the fibers. And that's how we understand as how they mechanically remodel the extracellular matrix. They also can digest it, they can secrete it too. So in order to understand the mechanical aspect, we built a elastic collagen network model. We took the experimentally available single fiber model and experimentally available bulk measurement. What's not known is a mesoscale cross-anchor structure and how many they are, where they are, so we fit for the network structure. And we can also have a finite quantum model that will and we got ambitious because we have the mechanics. We know the cell PCM interaction can be biomechanical. Can we also have a biochemical, biomechanical cell such that we just interact with everything biomechanically, realistically? It turned out that we could do some of this, but this part, the intercellular actin polymerization for biocell contraction was pretty difficult because it's a reaction diffusion equation with moving condition. So we proposed the scheme, and the student had graduated. But before we did all that, this framework can be used to calculate if you have a cell, it's the protruding front, the moving front, leading edge has the cell protruding ectomyosin protruding inside the cell, the membrane tension holding it back, and then there's adhesion to the substrate. And depending on the substrate curvature, this balance of force has slight angle, right? When you have to sum up all the forces, there's an angular component. So that in our mechanics calculation, full balance of force calculation, show that the cells on the concave surface will protrude more persistently, migrate more persistently than, for example, on this kind of curvature. And that's actually now being confirmed in the experiment that cells migrate very differently on such troughs than here. And which is highly relevant in vivo because in vivo, none of the cells is really walking on flat surfaces. Everything is curved, either inside or outside, some curved surfaces. And another piece of work that we did was intracellular, the cytoskeleton reorganization, that when you have a cell being pulled from outside, either this way or that way, the cytoskeleton, actin cytoskeleton reorganizes. So we did a little bit of that too. So we thought we really understood. Well, at least we understood a lot of the cell ECM interactions. Then this experiment came up, and this has been over 10 years old. We didn't understand it, and we still don't. Hemospheroid green fluorescent. It's not in matrigel, not in collagen, it's in matrigel because second harmonic takes hours to get all the collagen images. We want to see how really a much faster time scale. So we seeded the matrigel with fluorescent beads, and you can see how the beads are being pulled into the spheroid. Actually, before the cells start to move out. it takes hours for the cells to get familiar with the environment and then try to reach out. But from the very beginning, the bees are being pulled into the spheroid. And we don't know how, we don't know why. And again, yes, we can talk more. We have some hand waving.
Sorry,
Yeah.
Can you show it again?
Absolutely. I should
It's not
probably,
that obvious.
I should really make it loop. How do I video? I should play automatically and then loop. There we go. See the bees are zooming in. And then the cells start to go out.
So the tumor is in a gel and the red beans are frozen.
Yeah.
Colloids
Yeah.
in the
In the
gel.
gel, yeah,
So it's digesting the gel, I mean,
could be.
well, because
Okay.
you're putting things in, it has to,
Yes.
it has to go somewhere, but the
Yes.
gel has to be dissolved.
Well, the gel can be, my hypothesis is actually the gels are really floppy, disordered, fibers. Such that if I want to walk on such fiber, I have to tug it.
Yeah.
Until it's taut,
Yes,
and I can walk out.
Right.
So I have a ball of yarn, in my collection here. I could also be eating the yarn, I don't know.
Is there any accumulation of the gel? I mean, we're seeing in beads, but what about the gel itself?
I
Is
don't
it-
know, we can't image it yet. We couldn't, maybe now. But the whole stuff that. So, and we do have some analysis of these kind of beads and those kind of beads and the cells, so we can talk about that too. So this piece, we can quantify ECM remodeling, we can study the ECM mechanics and cell ECM mechanics, but there's a lot that we don't understand. We try to get a full mechanical story as well as the biomechanical story, but it's not quite there yet. And we told you about this collective relation. The observation is about 20 hours. What happened after a long time? In 2-3 years, the leaders separated out, remain leaders. And in 2 months, 1 to 2 months, the collective thinkers start to come up of the followers. The leaders remain leaders, followers can turn into leaders. And if you grow this in hypoxia, that's what happened. The leaders emerge in 2 days. So harsh environment, harsh conditions, stress leads to emergence of leaders. We don't know how. And we know the leader, follower, and parental cells are all different in shape, in genetic, in epigenetic, in single cell seq, you can see different states. Are they different states? So we need the follower sitting down here and parental maybe sitting up here that you have to travel upward in order to become the parental, or you can have 3 well structure that you can take the well in different ways such that you can be pushed one way or the other. We don't know. My hypothesis, again, this is all hypothesis, that hypoxia can induce VEGF, HIP1 alpha regulated VEGF dynamics, and VEGF binding can inhibit delta such that this large delta lateral inhibition can induce a delta rich cell, that's the leader. And then the lateral inhibition is such that this guy is the follower. Right?
Sorry, but for someone in a different field, it's very difficult to understand.
Sorry.
That can you say in a sort of
All right.
simplistic manner.
Okay, here. This finding the VEGF that's secreted by the liver cells or the in the environment, and this finding is known in angiogenesis to lead to the inhibition of more leader, to create more delta in the cell. Delta is my surface membrane protein, and you have my delta will interact with your arm. this interaction, this paint-out, shaping of paint, these two intracellular domain of notch, you have your notch breakout inside and such that it will go into the nucleus to regulate this intracellular. It inhibits the VEGF-R, the receptor. It promotes more notch. It inhibits delta. So my delta interact with your notch such that you will have more notch and less delta and less VEGF-R. And
So I have a question, so.
Okay.
No, it's okay, I'm understanding, so, but given the details that you have to go through to explain interaction, something that eventually move, in space outside. Why, so, is that people try to model this because, someone was asking before, so, but what's the possibility of measuring parameters, and then you said they're unlikely. So why do we go to that route since we have a few chances of measuring anything?
this is actually pretty well understood in angiogenesis. Like what's her name? Baker. No, not Ruth Baker. The other lady doing angiogenesis. Ellenberg? No. Ellenberg did angiogenesis. The very quantitative perfusion. She talked about leader follower dynamics. She had the leaders protruding all the phytophobia in zebrafish. Sorry, I'm blocking on the name, but this signaling network is actually quite well understood in angiogenesis. And I'm borrowing that for leader follower. This delta notch interaction is you could potentially measure all the genetic rates. So
But you understand my question? So
Yes.
if I'm looking at something at a much bigger scale, how relevant is it that I know exactly the parameters, assuming that I can measure it? I know
Right.
exactly those parameters. I can just
no,
also
but,
represent that interaction in a different way, for sure, no?
right. But in this context, I could also do qualitative understanding of the possibility of different types, steady states, such that remember in the beginning, I said the leader is high in jagged. In the low jagged, jagged is similar to delta. It will interact with your notch. So jagged actually trying to disrupt the notch delta interaction in that lateral inhibition. So with the jagged, high jagged, we can get a 3 steady state. This is almost similar to EMT and epithelial mesenchymal transition that used to be just 2 steady states. Now there are mathematically you can show there are intermediate steady states if you look at the topology of the impacted color.
I just wanted to ask, what was the connection with the EMP? If you, like in your leader follower, actually mesenchymal, it could be the intermediate ones.
Right, EMT in leader is more mesenchymal like, follower is more epithelial. All these cancers start out epithelial. So if leader is more mesenchymal like, it has to go through a EMT transition.
Okay.
And that E and M, two phenotypes, can maybe defined by this delta level, large delta level.
And I just wonder what where you can snail.
Yes,
And
yes,
other.
that's for the EMT, the classical EMT. But I'm trying to show here that I can define 2 phenotypes. These 2 phenotypes could correspond to leader and follower. But then because of the high jagged in leader, I could even have 3 phenotypes. And that intermediate phenotype, I have no idea what.
They do observe what's intermediate EM.
Could be, we don't know. We don't know. And then this is one hypothesis how leader could potentially emerge because of hypoxia and emerge faster. And then there's other hypotheses that leaders emerge. Hypoxia induce this caffeine interaction such that it inhibits the integrin binding such that it will be amoeboidal migration instead of mesenchymal migration. So different hypotheses. How do we test it? Can we link it to the 2-month versus 2 day time scale? How do we do that? I don't know. So I'm bringing more questions here, and I'm hoping for more ideas from you. So, okay. So collective conservation as a ecosystem point of view, that we are looking at ECM interactions, all those things put together as a social interaction network. We can find minimum conditions, we can find more mechanics, and the mechanics seem quite messy that we don't quite understand what's going on. And the hope is really to look at all the social interaction network and try to identify weak links to disrupt collective innovation instead of trying to kill the elusive leader cells. A leader emerges, may lead to I don't know whether intermediate phenotype plays any role in here and how to interpret it at all. So many unanswered questions, and I'm trying to bring here that anybody head into whichever point that we can talk more about. So our models are wrong, and I'm sure a lot of it is not even useful here. So I hope some of it may answer some questions, and we try to start with the simplest questions. Those are my students and collaborators at different aspects of the project. And if I have a few more minutes,
Yeah.
I would talk about, for the physicists in the audience, the topological defects and active matter in epithelium. So this is one of the earlier papers, well, not so early, papers about epithelium in Drosophila that leads to different alignments, leads to different stress points and topological defects. And we are trying to map that into retinal pigment epithelium in the eye. And this is something that I did a long time ago. We put two-dimensional bubbles, forms, perfect hexagon patterns, and then I let it flow around the obstacle. And that creates about these 5-7 pairs of defects, topological defects. And they are generated, annihilated, traveled, carried along, and all sorts of interesting things that I don't know how to analyze and how to model. And we also put previous piece of work, we put collagen in different channels, the different width, narrow ones, basically force alignment. And that when you see the cells in these different channels, because of the collagen alignment, they have very different protrusion dynamics and migration dynamics. So, and what else? I don't even remember what I put in here. Okay, actin cytoskeleton and myxobacteria. So here's actin cytoskeleton. We used medium mechanical dynamics of active networks from Papoyan lab at Maryland. And we looked at, without external force, the actin, myosin, and alpha actin, then they just collapse. And with external force that you pull it, they form these bundles. And you can change arbitrarily the actin concentration, myosin concentration, alpha actinin concentration, their turnover rate, their binding strength, their polymerization rate, etc., etc., etc. And it's a very flexible system that you can use to explore the remodeling dynamics. You can have them form this stress fiber and then you can move the beads, pull them, and contract them. You see how that changes the remodeling. And this is another area of my research, actively ongoing, based in eye diseases. We look into angiogenesis in eye development and in age-related macular degeneration and retinal pigment epithelium in the back of the retina, separating the densest blood vessel network and the photoreceptors is actually the key site for this disease. And it's a two-dimensional single layer of cells, and how they structure and how the different morphology, which you can see in the dissected eyes, in mouse eyes and human donor eyes, how that relates to age and how that relates to disease status. And we are interested in modeling the damage and repair. We're interested in eventually be able to indicate in vivo that if you go to see a clinic, eye clinic, and they now images your blood vessel, but they cannot image the cell pattern. And we are trying to bring together astronomy, astrophysics tools, both in optics and in big image analysis to try to look into the back of the eye. And another piece of work that relevant to collagen is liver fibrosis, is 3D deposition of collagen. And that fibrosis leads to the blood vessel stiffness change and blood flow dynamics change that can be measured non-invasively using ultrasound Doppler. And we have a technology that can measure ultrasound Doppler, use ultrasound Doppler, and then lead to different stages of fibrosis. Because fibrosis is actually in the early stages, all chronic liver diseases conditions lead to fibrosis. In the early stages, 1, 2, and early 3, it's reversible. You don't feel it, you don't know it. There's no reason for the doctor to try to image your lung or do a biopsy. The gold standard is a biopsy of your lung tissue. But as soon as you go through stage late 3, stage 4, it's cirrhosis and it's irreversible. So early detection is actually quite important, and if you change your drinking habit, if you don't drink alcohol, if you treat your hepatitis, you treat the underlying disease, you can reverse it. Anyways, so that was a technology, and happy to talk about it. And in the last few years, infectious disease. Infectious disease was the trend that the world focus. So we were looking at reversibility clearance of the immune. We were looking at chronic acute inflammation. Some of them lead to cancer and some of them don't. We're looking a little bit at the antibiotic resistance. We looked at COVID epidemic modeling. A co-evolution of epidemic and infodemic seemed very interesting. That SIS model for people carrying good information and thus behaving well, and versus people who are carrying bad information as not behaving, not following guidelines. This is the one that's spreading virus. But then for information, you can also be exposed to all your friends, all the people that are giving you bad information, you can be misinformed. Or if you are really sick now in the hospital, you now finally realize it is real pandemic. It's COVID-19, not a big flu, that you may behave better, right? So there's this cycle of information exchange versus viral infection. And then you can also choose to become vaccinated, and the vaccination carries the risk. And you have to do a calculation of risk between vaccination and infection. So this is the 5-dimensional model that's very rich in dynamics. You can have disease-free equilibrium, you can have coexistence, you can have simple oscillation, you can have mixed oscillations. And I don't have a good handle of this high-dimensional dynamics. Anybody who's good at math can help me out. I think that's a lot. Thank you.
So getting your first part on the summary model. So I think I missed this. What's the microscopic dynamics?
Microscopic dynamics of osmosis.
Yes.
It's the differential addition, the type dependent boundaries strength, coupling strength between the boundaries and with the geometric constraint. And then there's a bias in terms of copying in different directions.
And so what does the microscopic equation of wave motion like?
factor in terms of updating rules.
The safety side and this program and then go through that. and talk
Yeah.
about,
Yeah, So-
neighboring sites.
Neighboring sites, for example, I pick these three and I turn it into two. Now, cell number two grow a little bit, cell number three shrinks a little bit.
But is it physical dynamics? Right? Because this thing, like, for example, Metropolis Monte Carlo and Ising model, right?
Yeah.
You can do this, but that's not.
yeah, we do copying instead of swapping, but then, you calculate all the energy, and if it lowers the total energy, you accept it. If it, increase the total energy, you accept it with the Boltzmann. With this.
Yeah, but that not necessarily fulfills the equation for motion, right? I mean, that's just the way to reach some kind of equilibrium, right?
Yeah.
But
Yeah.
not necessarily using the following correct dynamics.
What do you mean by correct dynamics? This is a way to force the cells to jiggle and arrive at a lower energy
Okay,
configuration.
but assuming that there is some kind of physical material, right?
Yeah.
That has fulfilled some kind of equation for motion problem. Equation also, so friction times velocity physical force, right? In overcome limit or Newton equation. right?
Okay, but this. The cells are forcing this extreme. And they are moving, if that's the case, they are moving. So there's definitely energy dissipation because of this. Yes, energy. It's a, it's fluctuation. With dissipation, definitely dissipation.
Okay, the point is that
I don't.
even if I can use the energy and then differentiate it with X to some degree.
Right.
We get drive for moving certain direction. It only happen to move in that direction if you happen to take that speed and happen to take it.
But I do that over all the things, right?
Right, and you
And
have
that's repeated over and over, right?
Yes, but then there's no force to drive in a particular direction.
you can put a
But
gradient in
this
mu.
is the gradient that I'm forcing it.
But you're forcing it only if you happen to take a speed in that direction.
But they all get sampled all the time.
They get sample all the time, such that overall, over time, you will get a bias.
But
Yes,
But you have a gradient in mu, is that correct?
I have, this mu is either positive or negative as a constant. The gradient is in the external field that I give it.
Which term is the field?
C is the field.
Okay, so that just.
C is the external chemical field.
So C
right.
is the local value.
C is a local vector. Yeah. And I can set up as a gravitational field if I want to, right?
I liked how you went from here, if I remember right, towards trying to use learning techniques to see if you can, model the fluid like macroscopic behavior to some sort of, PDE. And when you say fluid, I can see how the motion looks very fluid-like, but I can also imagine differences. I mean, I imagine this blob of cells does it respect momentum conservation? Does it respect mass conservation? Because it can grow. So would it necessarily even lead to Navier-Stokes equation?
Don't know, but in here, I have not put in growth. I have not put in that. The simplest conditions, there's mass conservation.
Okay, so mass conservation is there. What about momentum?
I'm certainly not concerned with them, and that's no, you're not very small. The edge can roll out. That's
But
really not
the
considered.
chemical gradient forcing it to move, it's almost acting like a pressure, really.
So, I guess Jose is asking about momentum conservation.
Yeah.
So the
Is it
center
quite-
of mass has to be fixed.
Yeah. So, yeah,
It's
because like
driven, why would it fix?
I'm just saying that it could be something even more exciting than just Navier-Stokes describing it through the fluid, since there's more going on here. So
Right, so
yeah, that's like this. is-
talking about a necessary two equations. One is describing the front, the other is describing the dynamics of the leaders, because the leaders are the driving force
Yeah.
for such tips. Yeah.
No, so those is what I term the physics informed terms that we probably need to include. Sorry.
I think that's.
Right. So it's like those polarity in dynamic materials, right? You have additional physical property.
So in this video that you're showing, is it the number of cells is constant?
Yeah.
Okay. Have you considered doing like studies with or the like learning a particle based model using the sensors of the cellular pods? Collected, I guess the most differential equations that might gotten past there.
Learning those equations.
Yeah.
You
You could track out the centers of all these like clumps of things, and then use those trajectories to learn an ODE. Or
Isn't that similar to what we do? Also,
instead
instead
of
of looking at front,
you'll look at
all the cells.
yeah, I thought you were doing a PE. Yeah, yeah. And then the other question was you had early on, you showed two pictures of experimental data where there's different numbers of leaders, followers, and you got more or less growth. In those experimental conditions that they try many different ratios of leaders to followers.
So because leaders don't grow very well, the followers grow and then you almost always end up with like 50% of leaders. So you can take the leaders, inject into mice, they don't grow. You take the followers, inject into mice, they don't grow. You make a mixture of 1,000 of them can grow. And if you harvest the tumors after a while, it's like 1 to 5% of the. So there's the magic several percent minority leader or informed individuals that are losing them. So that's the also the magic number that I'm very good. In me and cousins and colony migration informed with the only needs to be a 50%. So I will accurately.
So in the phase space of your very first model,
Not my model. Yeah.
one
Yeah.
the real ones probably live in the parasitic in the mutualistic region because they need each other.
Right. Right. So that if you interrupt the mutualism through the branches, you can maybe cut down the inflammation and growth.
In the other part, the one with hypoxia, you showed two different experiments, one at 21%. The one at 21% eventually develop an hypoxic core, or because eventually
good question.
This fairly grows, you would expect the center to become
Good question.
hypoxial soon.
I'm not so sure. It could be, yeah, it could be 2 months. We have to calculate the size.
It's because of the size.
Yeah.
Because maybe the growth rate driver later on.
Yeah. Yeah. So hypoxia is a driver. We don't know how yet. That's a strong thing.
Okay. Thank you again.